Now we come to the most exciting part of this talk, which is the introduction of the Veneto inverted imaging platform. This is the result of many years of development from our microscopy division. The Veneto is a modular platform that can be used as a development environment or as an integrated systems platform for routine imaging experiments, or, as I'll show you, it can be both. This flexible platform can contain sophisticated two-photon or confocal systems and still allow multiple access ports that can be used for experimental optical systems without any possibility of disrupting the alignment or the functionality of the other optical modalities. It really is two microscopes in one. The core of the Veneto is its unique ability to have up to 7 optical paths that can be individually selected by the operator. This allows developers to share valuable table resources, such as lasers and modulators, between different imaging modalities and be able to quickly and accurately switch back and forth.
Now we will go through the major systems of the Veneto. As you can see, it is very much configured like a three-dimensional breadboard with some familiar-looking inverted components. The modular design allows full access to everything, including, and most importantly, the optics. Optics with various focal length as well as coatings can be inserted. However, the standard focal length tube lenses can always be quickly and accurately returned to their intended positions without any realignment. Let's start with the lower primary light path. This path can be used to introduce a laser scanning system for confocal or multiphoton imaging, or can be used for wide field or selective illumination. In the example here, we are showing a confocal module which relays light from a single mode fiber to our galvo resonant scan head. For the return beam, the light is descanned by the resonant galvo pair and then spectrally filtered before being spatially filtered by our motorized pinhole assembly. The light is then picked up on the multimode fiber and sent to the detection module, which we will be highlighting in the next slide. This is our multi-channel confocal PMT detection module. It can be easily configured from 1 to 4 detectors, and the dichroic and emission filters are housed in removable kinematic cubes. The module can be located on the table or neatly contained inside the microscope body. The light is efficiently coupled to this module from the pinhole using a large core multimode fiber. The second level of the Veneto is pre-configured as the reflected illumination module. However, for maximum flexibility, the illumination path can be removed and reconfigured using the breadboard pattern on the spine of the microscope. The light path shown is optimized for even fluorescent illumination from a liquid light guide or LED source. The fluorescent turret is fully motorized and contains spaces for 6 sets of excitation and emission filters. The design of the turret is such that once slid out from the microscope, the filters are easily accessed by removing the cover. The dichroics are held using small springs as to assure that the mounting does not produce any mechanical stress on the optical elements. The objective tier has 2 major sections. The front contains the objective turret, as well as internally a motorized selection device that allows the introduction of laser-based autofocus systems, or allows for a dichroic to provide a path to the non-descanned emission path. The turret is a very smooth-acting mechanism that allows for 5 M25 objectives to be installed. Attached to the back of the objective module is the non-descan detector module. This is used for collecting the emission signal from multiphoton excitation. It collects light at an angle of 13 degrees, which is essential for ultrasensitive detection deep from within thick samples. The module has 2 gas PMT modules and accepts a standard dichroic and emission filters, both of which are on magnetic mounts and can be easily changed by the user. Attached to the third tier is one of the most innovative parts of the Veneto. The Z-drive assembly is driven by a closed-loop direct drive motor assembly. The encoder is mounted on the turret so that the feedback is as close to the objective as is possible. This removes any backlash or hysteresis from the system, assuring accurate Z positioning down to 10 nanometers. Here's the cool part. Additionally, there's a 100 micron piezo that is integrated into the focus mechanism. That means that anywhere in the 15 millimeters of motorized travel, the user has access to 100 microns of 10 nanometer resolution rapid piezo stepping. This allows for rapid Z-sectioning with any of the attached objectives. With 100 nanometer steps, it's possible to image up to 15 Hz.
Now we move to the lower tier of the microscope, where we find the light path selector. This unique feature makes the flexibility of the microscope second to none. The light path selector is fully motorized and allows access to 4 ports, all of which have SM2 threads as well as 60mm cage interfaces. Each of the 3 positions on the selector platform can be easily set up with the included kinematic mount to direct the light from the microscope body to any of the 4 ports: right, center, left, and forward port. The motorized selector then can be moved to alternate between the preset ports. The front port can be used for an additional light path or the trinocular. The front port can be configured as an additional imaging port, or in the case of the system shown here, as the trinocular port for direct viewing. The trinoc also contains an additional camera port. The eyepieces contain a laser interlock, as well as positions to insert IR blocking filters for additional safety measures. The transillumination module also has some very unique features. The condenser control was designed with user ergonomics in mind. We have multiple condensers that can be inserted into the light path. The top end of the light path is removable and has standard cage and SM thread interfaces so that multiple different illumination or forward detection systems can be attached. The Veneto was designed to work with the MLS 203 stage, which was the industry first linear motor driven microscopy stage. With its silent, smooth motion, it is capable of high precision moves while still being able to move at a remarkable 250 millimeters per second. This makes the stage perfect for use for slides as well as 96 well plate formats. The control is via USB and/or with an ergonomically designed joystick. Now that we are familiar with the platform, I want to show 2 common configurations of the Veneto. First, which is pictured here, is a configuration that includes our galvo-resonant confocal with wide-field fluorescence. As you can see, the entire scanning and detection system is contained within the platform, and the external components are the fiber-coupled laser launch, the stage controller, and the LED illumination controllers. Towards the front of the table are the stage controllers as well as the automation controller for the microscope. This configuration is perfect for a multi-user facility or for a lab where routine work is expected, but the additional ports can be harnessed for optical systems development. This complete system highlights a multi-photon configuration which includes either resonant galvo or resonant galvo-galvo scanning with non-descan detection. On the table are the microscope control components from the previous configuration, as well as the Tiberius, our Ti-Sapphire tunable femtosecond laser. Between the laser and the microscope is our motorized variable attenuator, as well as a Pockle cell for laser control. The entire system is controlled by Thorlabs LS. Both previous systems are controlled by an extensive software package called Thorlabs LS. This robust package controls all of the Thorlabs imaging modalities in a single, easy-to-use platform. It has the ability to configure experiments to contain external triggering and automation within MATLAB, ImageJ, and an internal scripting interface. We provide free lifetime software updates and have also open-sourced the code for our software so that users can have full access to modify the software as needed for their individual use.